Here's what I want you to do is go to View. Go to, you should see a Venn diagram. Where is it? There it is. It's kind of in the middle, right below XY plot. Hit Venn diagram. Okay. First off, let's select off this. Select C. This is our time 0, right? So we have our 1,000 genes that are different at time 0. Now hold down your command and hit, or if you have a PC, I think it's Control. Select the other one. These two lists, this is basically what it's telling me. There's a lot of similarity here. 25% of both these top 1,000 genes are shared. There are some genes that change that are different between the two regardless of differentiation state. We also find there's a lot of genes that are different too. About 75% of those top 1,000 aren't in each other's list. Now let's put the other one in. So Control or Command or whatever you got, hit the other one. So this is E, and we can call this anything we want. So we can manipulate this. Let's do this. So click on, you can just click on D, and it should bring up this config Venn diagram. Everybody got that? First off, those numbers are incredibly small, and I hate that. So let's go for label font, let's go, I don't know, 24. Font size for the name, maybe go 20. No, 22. Why not? Now here we can put what these different things are. So C was day 0. D was day 5, right? CD. Right? And E was day, let me put a star cell type. Hit OK. There we go. I use Venn diagrams a lot. And as you can tell, with so many genes different, basically what I want to do is I want to get a smaller group. The smaller the group, the better chance I have of finding something overrepresented in it, like a pathway. At the same time, I don't want it too small. And what happens is, as you do this more and more, you just kind of get this kind of natural feeling of like where the sweet spot is. I'm going to tell you right now, it's probably around 300 genes to about 1,200 genes is my sweet spot. Another way to think of it is, usually if you're looking at more than 5% of all of your genes that you're surveying, it's probably not a good idea. Anything less than 5% of the total genes. So in our instance, we've got, what, 35,000? So 5% of that would be, what, 1,500? Is that right? Did I do that right? 1,700? I thought you were bioengineers. 1,700, right? Somebody say yes. Okay, good. So if I got a gene list of more than 1,700 genes, I'm probably not going to do well. What happens is I'm looking at too many genes, I can't find those overrepresented items. And you're going to see when we do Ingenuity, like you'll see where that applies. Okay. As I look at this, though, I'm interested in these genes, but here's the deal: is I want genes that are changed due in day 5 and day 0, but also have this day and cell type effect. Here's how you get those genes. I can select one of those, so I can select the middle. These 25 genes would be very interesting as well. And actually, if I was working at Collaborator and I gave him these 25 genes, he'd be ecstatic. He'd be like, oh my gosh, 25 genes. They can process 25 genes. Sort of. Honestly, most of the people I collaborate with, they're looking for one gene, right? Like, what's the answer? And I'm like, this is not an answer, it's a story. But they don't like that. They want a target. They want something. If I was looking at a target for this, I'd probably find in this 25,000. Let's make a list of that. Everything that's in all. So I right-click as it's selected, and I say, create list from highlighted regions. Boink. Here are all those genes. This is probably a very good list to go through. Like I said, if I gave this list to my collaborator, he'd be very excited. I'm seeing a lot of microRNAs as well. And researchers love microRNAs because they're easy to add to the system. If you pull up a microRNA for them, they're like, cool, we can do that easily experimentally. That's another thing I want you to keep in mind is the targets you give, the targets you produce from this have to be practical as well, right? You know, I can't, you know, if I do a cancer study and I go, hey, the answer is P53, my investigator is going to go, well, great, you know, we've known this forever and we can't replicate P53, so your information is useless. But if I say, hey, it's this other thing that interacts with P53 that's probably modulating it, then they get excited, right? As we get into the biology, the biological interpretation, you're going to kind of see this. Let's go back to our Venn diagram. I want to make another Venn diagram. Or I mean, not a Venn, I want to make another gene list. So for me, what I want to do is I want to look at all of those genes. Actually, I want to make 3 lists. I want all those genes that are changed at day 0 and day 5 significantly in the top 1,000. So I select those 2 that are basically common to this, and then I can go say create list from highlighted region. There it goes. Okay. And again, and here's another thing. I like things that are different at day 0 or day 5, but also have an effect as far as the differentiation state of these cells. I think that these genes might be relevant, so I can actually select these as well. So I select 336, 25, and 237, right-click, create list from highlighted regions. Dope. This is my gene list that we're going to use. So the next time we actually meet for class, what we're going to do is I'm going to go through here. We're going to take these lists and we're going to upload them into Ingenuity Pathway Analysis. And this is basically a data set, the very well curated, extensive data set that has mined all the scientific literature for relationships, for pathways, for signaling networks, for upstream regulators. Basically, what I'm going to do is I'm going to throw these genes in and I'm going to say, is anything overrepresented here? Like, say, you know, are these 6 in the same pathway? If they are, and that's, you know, the probability of getting that by random chance is very rare, that's something I want to look at. So let's save these. You can save them on your own, I guess. Wait, this one was, I forgot what this one is. File, save as text. Again, put everything in that title. So it'd be, usually I go ANOVA because that's what I did. ANOVA, common, day 0, day 5, and day, I'm not labeling these very well. I'm kind of off today. And cell type. Okay.